Across every orchard, an invisible cement helps each ripening fruit hold its shape. That cement is pectin, woven through the thin walls of countless plant cells. It gathers richly in apple peels, citrus rinds, berries, and purple grape skins. Beneath the microscope, pectin fills the narrow spaces between neighboring plant rooms. It gives crispness to fruit and quiet firmness to tender growing leaves. In jams, this same molecule helps sweetness set into a trembling gel. Yet the same strength can become a stubborn lock around useful liquids. Press an apple and pectin traps bright juice inside its pulpy walls. Crush grapes, and pectin can hold haze inside the young wine. In citrus peel, it binds water, perfume, and bitter compounds together. For centuries, people cooked, pressed, strained, and waited for these locks to loosen. But in forests and orchards, the answer was already alive and quietly moving. Fallen fruit softens because unseen organisms begin a slow, careful demolition. Fungi send fine threads through the flesh, searching for available sugars. Bacteria gather in bruised places, opening walls one strand at a time. Ripening fruit also carries its own quiet tools for controlled seasonal softening. Together, these natural workers reveal the talent we now call pectinase. Pectinase is not one blade, but a family of small matching keys. Each key recognizes a different part of pectin's tangled, branching chain. Before anyone named the enzyme, chemists first had to meet pectin itself. In 1825, Henri Braconnot examined plant juices in his Nancy, France laboratory. From apples, pears, and roots, he isolated a substance that formed gels. He named it pectin, from a Greek word for something congealed. Suddenly, jams, jellies, and fruit firmness had a new material history. Later 19th-century chemists separated pectin, pectic acid, and stubborn insoluble protopectin. Their notebook suggested that plant firmness was chemistry, not only kitchen mystery. Meanwhile, rotting fruit kept offering a more living and persuasive clue. Healthy tissue stood firm. Infected tissue collapsed around spreading fungal growth. Plant pathologists learned that microbes were not merely staining or poisoning fruit. They were releasing hidden substances that loosened plant walls from within. By the early 20th century, scientists could extract these softening substances from microbes. They watched mold cultures turn firm, plant pieces into flowing pulp. Useful strains were found among Aspergillus molds, common companions of decay. In fruit laboratories, pectinase became a practical helper for stubborn, cloudy juice. During the 1930s, enzyme preparations entered commercial juice and wine work in Europe. The industrial story began modestly, with selected molds and careful patient observation. Apple juice could clear more easily, and presses released liquid more readily. Winemakers found gentler extraction from grape skins, berries, and pressed pomace. A cloudy craft had gained a biological assistant from the compost world. Pectinase then moved from cellar trials into wider fruit processing practice. To understand that help, imagine a plant wall made of sticky ropes. Pectinase approaches those ropes without flame, brute force, or violent chemistry. One member clips the long pectin backbone into shorter, drifting pieces. Another loosens side branches that hold neighboring strands close together. Another opens tiny chemical clasps that make the chain especially clingy. The wall relaxes, and trapped water begins to move outward. Juice runs more freely through the press cloth and collecting channels. Floating haze particles lose their pectin coats and slowly settle downward. Aromas leave fruit pulp with less struggle and less discarded material behind. The enzyme is never a hammer, it is patient, selective biological lockpicking. In everyday kitchens, pectinase hides behind clearer apple and berry juices. In wineries, it helps fruit express color, aroma, and natural brightness. In berry processing, it can release deep pigments from fragile skins. In coffee processing, it helps loosen the slippery pulp around beans. In cocoa fermentation, it helps sweet surrounding pulp drain from precious beans. In citrus work, it helps peel residues become useful, flowing materials. In tomato and vegetable processing, it can improve the movement of thick pulp. In textile preparation, pectinase can soften plant fibers with gentler conditions. Linen, hemp, and ramie release gummy barriers with less severity. In paper and pulp work, it supports cleaner separation of fibers. In plant-based feed research, it helps nutrients emerge from cell walls. In waste streams, it can help recover value from fruit residues. Each use begins with the same ancient orchard problem returning again. Plants build strength by tying their cells closely together in moist walls. Pectinase answers by untying those bonds, not by destroying everything nearby. Its story bridges Braconnot's glassware and today's quieter circular biorefineries. It also bridges decay under leaves and industry seeking gentler tools. As fruit falls, nature shows that nothing is truly finished or useless. Pectinase reminds us that green technology often begins with listening carefully. The future may depend on such small, patient acts of release. At Enzymes Bio, we're a small team of scientists and biotech enthusiasts. We bring the world's enzymes onto one platform, so startups and labs skip the procurement maze. A greener chemistry for the years ahead. Learn more at Enzymes Bio.